Welcome to Inside Pediatrics with Dr. Chadia Jbeily, pediatrician with medical training from St. Joseph University in Beirut and Université Paris Cité in France. In episode 1, we will explore the antimicrobial resistance. The first question: when the antimicrobial resistance emerged?
In 1928, Alexander Fleming left a petri dish of Staph aureus uncovered, and 2 days later, he found that the mold contamination had killed the staph. He identified that the fungus Penicillium notatum killed the colonies of staph. Later, Sir Chain and Florey extracted the penicillin from Penicillium notatum, and so penicillin became available and saved millions of lives during World War II. Fleming warned about the emergence of antimicrobial resistance if antibiotics were ever misused or overused. In 1942, we saw the emergence of Staph aureus resistant to penicillin, and in 1945, Fleming, Chain, and Florey won the Nobel Prize in Medicine.
The second question: what are the main mechanisms of antimicrobial resistance?
Some bacteria are naturally resistant to antibiotics. It's just part of who they are. Take the gram-negative bacteria, for example. Their cell wall simply doesn't let certain drugs through. Then there is Pseudomonas. They actually pump some antibiotics out using what we call active exporters. And Mycoplasma, they don't have a cell wall, so some antibiotics just don't work on them. Now, other bacteria don't start off resistant, but they can become resistant over time. How? Well, 2 main ways. First, they can mutate. Basically, they change their genetic material, and that new resistant trait gets passed down to their offspring. That's what we call vertical transmission. Or they can acquire resistance from their surroundings. That's horizontal gene transfer. This usually happens through little bits of DNA called plasmids, which are circular pieces of genetic material floating around in the environment. Bacteria can pick these up in a few different ways. They might absorb DNA directly from the environment, that's called transformation, or they might get it through a virus in a process called transduction. And finally, they can transfer DNA directly from one bacteria to another, that's conjugation. You'll see this, for example, with E. coli and Salmonella, which belong to the same species group. Then there are the acquired mechanisms of resistance, where bacteria actually change how they handle or respond to drugs. They might reduce how much of the drug gets in, or pump it back out faster, or they might destroy the drug completely with an enzyme. Take E. coli producing SBL or extended spectrum beta-lactamase. This enzyme inactivates penicillin and cephalosporins. Some bacteria even produce MC enzymes, which break down not just penicillins and cephalosporins, but also third-generation cephalosporins, even those combined with beta-lactamase inhibitors. And then there are the carbapenemase enzymes that can inactivate the carbapenem. Another way bacteria resist antibiotics is by modifying the drug's target, so the antibiotic can no longer bind where it should. That's exactly what happens in MRSA, or methicillin-resistant Staphylococcus aureus. These bacteria have altered their penicillin binding protein, which makes them resistant to methicillin and in fact to all beta-lactam. And sometimes, instead of modifying the target, bacteria protect it, shielding it so the antibiotic can't reach it effectively.
Question: is the antimicrobial resistance well monitored, and how?
Yes, by the World Health Organization, by the European Reference Lab for Antimicrobial Resistance in Denmark, with Dr. Lina Cavaco, and by all the laboratories that work together as part of the international network. Antimicrobial resistance, or AMR, is monitored through what we call antimicrobial susceptibility testing, abbreviated as AST. When it comes to methodology, there are two major sets of international guidelines: the Clinical Laboratory Standards Institute, CLSI, and the European Committee on Antimicrobial Susceptibility Testing, EUCAST. Both are widely used across the world. We can perform AST in two main ways, qualitatively or quantitatively. Let's start with the qualitative method. That's the classic Kirby-Bauer test, which uses a petri dish filled with agar. There are strict standards for the size of the bacterial inoculum, the pH of the medium, the incubation time, usually 24 to 48 hours, and the temperature. Small paper discs containing different antibiotics are placed on the agar. After incubation, the antibiotic diffuses into the medium and we measure the diameters of the clear zones around each disc. Those are the inhibition zones. We then compare these diameters to reference tables to determine whether the bacteria is resistant, susceptible, or intermediate to that particular antibiotic. Now, for the quantitative approach, we determine what's called the minimum inhibitory concentration, or MIC. This is the lowest concentration of an antibiotic that prevents visible bacterial growth. There are several ways to determine the MIC. We have the macrobroth dilution. We use test tubes containing serial dilutions of the antibiotic in broth. After adding a standard amount of bacteria and incubating for about 20 hours, we check which tube shows no growth. That's our MIC. And we have the microbroth dilution. Here we use 96 well microtiter plates, each well containing a different concentration of the antibiotic. And we have the agar dilution. In this method, the antibiotic is mixed directly into agar at different concentrations, and the MIC is the lowest concentration plate that shows no bacterial growth. In addition to these traditional methods, we now have molecular-based techniques for AST. These include polymerase chain reaction, PCR, which detects specific resistant genes, for instance, MECA for MRSA, or genes responsible for ESBL production. And even more advanced is whole genome sequencing, which can identify all known resistant genes and mutations across the entire bacterial genome. There is an excellent platform called MyDB Finder where microbiologists and clinicians can upload the microorganisms they are studying to explore their genetic resistance profiles. Through these methods, we can build comprehensive data on the resistant bacteria and the genes that make them so.
The fourth question: how can we contain the AMR in pediatrics?
We have three tools: the hygiene, the vaccines, and the responsible management of antibiotics. If prescribed, we ensure proper dosage and schedule adherence and the time span of the treatment. Let's talk about the antibioprophylaxis in pediatrics. If we have a case of pertussis, we give the context antibiotics for 1 week. If we have a case of meningitis by Haemophilus influenza, the context will be given antibiotics for 4 days. If it's Neisseria meningitidis, the context will be given antibiotics for 2 days. In the curative antibiotherapy, if we have a case of impetigo with less than 5 lesions, we give cutaneous antibiotic. If we have cellulitis, 7 days. Bacterial infection, certain Salmonella, Campylobacter, Shigella, Yersinia, 5 days. If we have a case of tonsillitis, strepto infection, 6 days. If we have a case of otitis media less than 2 years, 10 days. Ethmoiditis sinusitis, also 10 days. Rhinopharyngitis, laryngitis, tonsillitis with strep test negative, we don't give antibiotics. Community-acquired pneumonia, it's 7 days. Atypical community pneumonia, 10 days. Bronchitis, mainly viral. Certain cases that last more than 3 weeks, we give antibiotics for 10 days. Pyelonephritis, 10 days. Cystitis, 5 days. Arthritis, 2 weeks. And osteomyelitis, 3 weeks.
Question: what were the past, actual and future actions to counter the AMR?
In the 1940s, we saw a wave of new antibiotic discoveries, many of them coming from fungi. Then in the 60s came the first generation cephalosporins and ampicillin, the first broad-spectrum penicillin. By the 70s, we had second-generation cephalosporins offering broader coverage and better activity. In the 80s, things accelerated. We saw the third-generation cephalosporins, the rise of broad beta-lactamase inhibitors combined with aminopenicillins, and the introduction of quinolone, which were purely synthetic. We also had glycopeptides, key antibiotics for resistant infections. Then came the 90s with azithromycin, an antibiotic that became a global favorite for its wide use and tolerance. From the year 2000 until today, 25 new antibiotics have been approved. In 2000, the World Health Organization launched a global strategy to contain antimicrobial resistance, urging countries to create national action plans that link both human and animal health sectors. Collaboration with the G7 nations soon followed, marking an important step forward. And today, research and clinical trials continue. With artificial intelligence, we can now analyze thousands of chemical structures and predict which ones might have antibacterial activity, especially against resistant strains. So let's keep this collaboration alive between microbiologists, physicians, and the World Health Organization to promote the responsible use of antibiotics and to keep innovating in this vital field. Thank you for listening.